A very good morning. So today we are starting with the structure of the body wall of Sycon. This particular diagram of Sycon shows the central spongiocele, which is the central cavity, and it is showing an osculum from which the water is going out of the body of the Sycon. So that we have discussed in the very first video of Sycon. So here we are to talk about the body wall. Now, can you see this particular yellow part? So if I zoom in into this particular part, so this part is the body wall. Here, the body wall, it consists of three distinct layers. So here you can see a light blue colored line, then a yellow area, then you can see a violet area. So that means the body wall of the Sycon has got three basic layers. So the outermost layer is the pinnacoderm. This is the pinnacoderm. So the picture on the right shows the three basic layers of the body wall. This is the first layer, which is the light blue colored layer, and it is the pinnacoderm. And it is composed of flattened cells which are called as pinacocytes. So this is the outermost first layer. Now the innermost layer, it is the violet layer, and here it is represented by QNO Sites. So these are the round cells with some collar-like structures with flagella. They are all the QNO Sites, and this layer is called as Choanoderm. This is the innermost layer. So these are the two basic body wall layers, which are the actual layers of the Sycon. The third layer, which is the yellow layer over here, this is called as the mesohyal or mesoglia. So this is the third layer which is secreted by the pinnacoderm. So this is not the actual body layer. If we go scientifically, the sponges are diploblastic in nature. That means they have got two body layers only. But the central layer, which is also called as mesohyal or mesoglia, it is very important in function. Now this is a gelatinous matrix-like layer which has got number of cells. So it is having different kinds of spicules also. So the cells present in this particular layer include: number one, Archaea Sites. These are the undifferentiated cells which are present in the mesoglia, and they are totipotent in nature. So these cells can make any kind of a cell in the body of the Sycon. So Archaea Sites are very important cells in regeneration, and they can form even germ cells, including ova and sperms. Number two, these are the colon sites. Colon sites, they are pseudopodial cells, and they are star-shaped also, having long processes or dendrite-like structures as we have in the neurons. So some scientists, they also think that they are working as neurons in Sycon. Number three, these are chromocytes. So these are pigmented cells which are present in the mesoglia, but they are having some kind of pseudopodial-like structures. So they are also a type of amoebocyte. Number four, thesiocytes. Thesiocytes, they are pseudopodial amoeboid cells which are storing food. So they help in storing of food. Number fifth, myocytes. So these are contractile muscle cells which are present around the ostia and osculum. Number six, these are scleroblasts. Scleroblasts are the cells which can form spicules. So we can say these are spicule forming cells. They are further of three types. First is calcoblasts, which are forming calcareous spicules. The silicoblasts, they are forming the siliceous spicules, and spongioblasts, which are forming the spongin fibers in the mesoglia. Next are the gland cells. So these are secreting some secretions which is particularly lime. So these secrete lime. Next are the germ cells, which include the ova and the sperms, and these are also present in the mesoglia, and they are formed from archaeocytes. So these are the types of the cells which are present in the mesoglia. So that's why this gelatinous layer, which is not the true body layer of the Sycon, is very important. Now talking something about pinacoderm, this is the outermost layer of the body of the Sycon. Here it is shown in blue color, the light blue color. So here it is showing the flattened pinacocyte. So these are flat cells, and the most important feature in pinacocytes is that they can contract. So they are muscular cells, and they can contract the body of the Sycon to a significant magnitude. Now, something about the innermost layer, which is called as the choanoderm. So here it is shown in violet color. So these are the choanocytes. Now we are taking up a single choanocyte cell. Now this particular diagram shows a single choanocyte. Now here you can see it has got a nucleus. It has got a collar. Now this collar is formed of near about 20 microfilaments. So these are the microvilli which are present in between the collar. It has got a single flagellum, and there are two other structures which are called as rhizoplast and blepharoplast. So that will be seen in the next diagram, I think. This is another diagram which is showing a single choanocyte. Now here you can see these are the protoplasmic processes which are embedding this particular choanocyte into the mesoglia, the yellow layer. Now this is the nucleus. Now here is a dot-like structure which is called as the blepharoplast. So this black structure is called as the blepharoplast, and it has been seen that blepharoplast is in connection with the nuclear membrane. So there can be some connection in between the flagellum and the nucleus. There is another thread-like structure which is called as a rhizoplast. Now this rhizoplast is connecting the blepharoplast with the basal granule. So this is the basal body which is present at the base of the flagellum. So every cilium or every flagellum has got a basal body at the base. So rhizoplast is connecting the blepharoplast with the basal granule or the basal body. So here scientists they think that the flagellum is in connection with the nucleus with the help of blepharoplast and rhizoplast. So there is nothing very clear about this, that how the movement of the flagellum is controlled, but scientists think that it is under the control of the nucleus with this particular structure. Now, after the structure of the choanocyte, now something about the functions of this particular cell. So this particular cell is very important in function, as it can perform feeding. So it feeds, it takes food, it can make food vacuoles. Next thing is this particular cell, it helps in reproduction also. So these are very important cells. And number 3, The whole water current that we have discussed in the canal system, the whole water current is made by these choanocytes. Now, first of all, I will be taking up the feeding mechanism in choanocytes. Now here in this diagram, what has been shown is the movement of the flagellum and the water current formed with the movement of this particular flagellum in this choanocyte. Now, whenever this flagellum moves, it forms a water current which is on this direction. So water enters the collar from this side and it passes away directly in the direction of the flagellum. So when the water it moves from this side to the collar, it takes up the food along with it, and the food is filtered in between the microvilli of the collar at this particular site. So Sycon is also a filter feeder because it is filtering the food with the help of the collar. In this diagram, you can very well see the movement of the food particles along with the water. So the food particles now they are being filtered in the collar, and after filtration they are being put into the food vacuoles. So this is a single food vacuole. Now the process of digestion will take place inside the body of the choanocyte. Now here the lysosome will attach to this food vacuole. It will digest the food as we have discussed in the case of paramecium. So this digestion is partial. This is not the full digestion. So in the choanocyte the digestion is partial, and this partially digested food is taken out into the amoebocytes which are present in the mesoglea. So here I have demarcated the mesoglea. So on the right side of this black line is the mesoglea, the yellow layer. You can see the yellow layer over here also. So this is one amoebocyte which is present in mesoglea. So the partially digested food is taken by the amoebocytes, and here the full digestion occurs, and this food is stored in the amoebocytes, and the food storing amoebocyte is called as zoocyte, and this zoocyte will remain in the mesoglea, and whenever required this food will be taken up from the zoocyte and will be used for making energy. Now there is a question that how the food particles they reached the choanocytes. This is a very important question, that how food reaches up to the choanocytes. Now I am answering to this particular question. Can you recall this particular diagram? Yes, this is of the canal system. So here this place is called as ostium. This is the pore from where the water entered into the body of the Sycon. So from the ostium the water entered into the incurrent canals, and along with the water the food particles are also coming inside. Now the food particles along with the water they have reached the ICC. From here they go to the radial canals. Now the water has reached radial canals. So here you can see the radial canals are lined by choanocytes. So these are the choanocytes. The food has reached the choanocytes. Now these choanocytes they will ingest the food by filtering out, and the filtered food will be taken inside the choanocyte. So this is the nutrition in Sycon. Now we will be taking up the second part of choanocyte. This is the role of choanocytes in reproduction. So choanocytes are very important cells. So first of all we should talk about the formation of the gametes in So here we have special cells which are called as Archaea Sites. So these are undifferentiated cells, and I have told you earlier that these can form any type of cell in the body of the Sycon. So the sperms and ova, they are also formed by archaeocytes. Suppose these archaeocytes have formed sperms and ova. Now these sperms and ova, they are present in the centermost layer, which is called as the mesoglia. So these are present in the yellow layer, which is the mesoglia, the central layer of the sponge. Now from here, these sperms will go out of the body of the Sycon. Now the sperms, they go out of the mesoglia into the central spongiocele. So in this diagram, the blue line shows the exit of the sperms from the body of the Sycon. Now suppose this is the Sycon number 1. So it has released the sperms out in the water. Now this is the sponge number 2. Now here it has been shown that the sperms of first sponge, they reach the ostium of the second sponge. So they will enter into the ostium along with the water. They will reach the incurrent canal, then they will go to the radial canals, and the radial canals are lined by choanocytes. So the sperms, now they have reached the choanocytes. Now the sperms of first sponge they have reached the choanocytes of the sponge number 2, and this is the ovum of 2. So this is the ovum of the second sponge. Now the sperms with some chemotaxis, they will enter that particular choanocyte which is very close to that ovum. So these sperms will enter into this particular choanocyte. So it is reaching the choanocyte number 4 because it is very close to the ovum. So here the head of the sperm, it enters into the choanocyte, and the tail, it remains outside. So here you can see the head has entered the choanocyte number 4. After the entry of the sperm head into the choanocyte number 4, it loses its flagellum and it loses its collar, and this choanocyte becomes round, and this rounded structure, the rounded choanocyte, is called as the carrier cell. So this is the carrier cell which will carry the head of the sperm to the ovum. So this carrier cell fuses with the ovum, and the sperm head is transferred to the reception cone of the ovum. So here the fertilization takes place and a zygote is formed in the very central layer of mesoglia. So here you can see the formation of zygote, and after some time the choanocyte number 4, it is replenished. It is formed by some archaeocyte and it replenishes. So thus we can see that choanocytes, they are very important cells which are helping in reproduction. So if there is no choanocyte, so the sperms, they were unable to enter the mesoglia. So here choanocytes, they are forming the carrier cells which carry the head of the sperm to the egg. So here the fertilization process is internal because it is going on inside the body of the Sycon number 2, and it is cross because the sperms which are coming inside, they are the sperms of first sponge. So the fertilization is cross and internal. So that was the function of the choanocyte. So students, that was all about the body wall and choanocytes.